Hi, everyone. So my name is Chi Hong. Thank you for joining me today for the topic and sharing of sample preparation for Sanger sequencing. Okay, so first of all, I would like to show you some agenda of today's sharing. So at the very first part, we will actually share on why DNA sequencing, and then after that, how does sequencing actually works, what is really happening, because a lot of time we actually outsource the sequencing, and then so I'll give you a little bit more information on actually what happens in the sequencing house. And next, I will share with you some sequencing result, some commonly see problem like some unsatisfactory sequencing result, and what are the most common cause of those results. Next, I shall go on with sample preparation or some of the good practice of to actually to clean up the PCR products, plasmid, how to actually send a good quality sample for the sequencing house to process and give you, and in turns give you a good sequencing result. So next will be some tip and tricks to actually give a better quality sample to send to the sequencing house. And finally, I will end with some advancement of the DNA sequencing, just a short slide on the advancement of DNA sequencing technology. Okay, so let's move on to the first topic of today, which is why DNA sequencing. So DNA sequencing by definition is actually quite straightforward. Is to actually we have a DNA fragment, double-stranded most of the time, and we would like to know exactly each base, what are the base. So as we already know, we have like A, adenine, then cytosine, guanine, and thymidine. So we like to know in one particular position from start until the end, what is the sequence of this particular bases. Okay. So originally back in 2003, so when the United States, they actually initiated the Human Genome Project, and they would like to sequence the full genome of a human. At that time, back in 2003, they are actually using the original DNA sequencing techniques, which is what I'm going to cover today, aka also known as the Sanger DNA sequencing technology. So at the time, this technology was used. And more recently, when we have the outbreak, okay, we still using DNA sequencing technology, but the more advanced version, okay. Although Sanger sequencing still can handle it, but because of the high throughput, high volume, then maybe the next generation sequencing is a better choice. Okay, so what is DNA? So DNA is actually the genetic materials that hold information. And when this information is required, it will be then translated into protein. And protein is the one that will be carrying out the job. Okay. So this is the just to show you that the relationship between DNA and protein. And the reason why is that whenever we talk about gene or a DNA sequence of a gene, we will talk about the nucleotide base, which is the A, T, C, and G. This is actually an example sequence of the insulin gene. Okay, so we have the start codon, and then at the end there we have the stop codon. And then as you can see here, 3 nucleotide will be form a codon, and one codon will be code for one amino acid. So by looking through the amino acid table, we can actually translate the DNA sequence into the amino acid sequence. So the up there is the DNA, the down there is actually the amino acid. Okay, so DNA sequencing technology is very important when we want to investigate whether there are any mutation or not. So we are doing some genotyping. So when we talk about, we want to determine the genotype of a sequence of an organism or a gene or something like that, we're actually checking the DNA sequence. Okay, If let's say, for example, there is a mutation happen in this particular location. So originally, let's say the sequence of the insulin gene is TGC, and there is a single nucleotide polymorphism. The T base here has been mutated into a G. So therefore, instead of TGC, now we have a mutated base GGC. So when we check through the amino acids table, you can see that TGC originally code for cysteine. Okay. However, when the mutation causing the T to become GGC, it will actually become a glycine. Okay, so we know that mutation will cause this, and when we go for DNA sequencing, we know that, oh, there's a SNP, there's a mutation. So what's the significance of this particular mutation? Okay, so the effect is that when we have the protein coded based on this particular DNA sequence, one is with the original wild type, and the second one is with the mutation, with the glycine. Okay, so the important thing here is that originally the cysteine will form a disulfide bond together with another part of the insulin chain. Okay, then you will actually stabilize the whole structure of the insulin and make it functional and become a functional unit, carry out the job of insulin, what insulin is supposed to do. However, if this particular cysteine here, okay, has been changed to become a glycine, okay, because of the mutation of one single base. Okay, so the chain that you see here will no longer be formed. And we lost this particular chain. Sometimes it's actually an important chain that to retain the structure, it might cause the protein reduce in terms of functionality. So this is the importance of why we are doing sequencing to determine mutation and how it actually affects the protein and how the protein carry out their tasks. Okay, so next, I shall share with you some DNA sequencing application that is commonly used. So normally in clinical research of diagnosis, we can use DNA sequencing technology for genotyping or mutation study. So this is useful whenever we want to try to look at hereditary disease such as hemophilia or thalassemia. And then also not to forget cancer or tumor genotyping. Okay, for example, leukemia that has a lot of fusion gene and mutation happening. Okay. So in addition to cancer and hereditary disease, we can also use DNA sequencing to look for antimicrobial resistant gene. So when we talk about hospital, we talk about the communicable diseases. So we have bacterial infection, and then the problem of those pathogen bacteria in the hospital, they a lot of times they already acquire some antibiotic resistant gene. So this is important if, let's say, those bacteria that causing infection to those patients in the hospital, they are very resistant to the drugs that we actually the doctor give up for the patient to heal, okay, to recover. However, if we manage to detect all these antimicrobial gene, we know which drug to choose to cure like disease like sepsis, yeah, or bacterial infection. Okay. So next, in addition to those clinical setting research and diagnosis, DNA sequencing can be used in the field of gene discovery and recombination technology. Okay. So when we talk about gene discovery in agriculture, we might need do some transgene to actually transform or to transfer gene that confers pest resistance or to produce higher fruit, to give more fruit, bigger fruit, or faster growth to those crops or any farm plant. Okay. So in addition, when we want to look for those new strain of bacteria, okay, those new strain of bacteria in the not so well-known area, for example, in the Arctic, Antarctic, or let's say some swamp area, those bacteria might carry some unique gene or enzyme that enable those bacteria to survive in those unique or niche environment. Okay, so this kind of enzyme might be useful to be engineered and then to become recombinant protein to be used as bioprocessing enzyme and later on for commercial use. So a lot of time, some of the example will be some protease from bacteria that enable to speed up some production in the factory. So certain step in the production of factory needs certain enzyme to speed up the process. Last but not least, DNA sequencing can be used for epigenetic study. And when we talk about DNA epigenetic study, we are talking about DNA methylation status of the DNA. So if the gene has been methylated, it might not be able to express and causing the turn off of the gene. And this sometimes can lead to like certain diseases. Okay, so when we move on from the introduction on some of the application of DNA sequencing, now I will proceed to how does Sanger sequencing actually happen? How does it work? Okay, so the first generation of DNA sequencing, which will be the focus of today, is actually called Sanger sequencing. Okay, so Sanger actually is named by the person who developed it, which is actually the Frederick Sanger. Okay, so this is this person, Professor Sanger, showing here. Okay, this particular technique, also known as chain termination sequencing, because it will actually cause the termination of the DNA strand. So it means that no base can be added after this particular base. In the original techniques, they are using the radioactive base, but as the technology advances, we are currently using fluorescent dye instead of radioactive labeling. Okay, here I'm showing the steps involved in Sanger sequencing. Okay. So the first step of Sanger sequencing, after we send to the sequencing house, of course, we will perform the quality check. So once the quality check show that the product is good enough to move on to perform sequencing, so the first step, we will actually perform what we call a cycle sequencing reaction. Okay, so this one is showing you the cycle sequencing reaction. So we add some reagent to produce the chain, the new strand of DNA. Next, after the production of all the different sizes of the new of DNA, we will proceed to dye terminator removal. Basically, it's a cleanup step to remove excess reagent from the first step. Next, when we have the pure DNA strand newly synthesized, okay, we will now separate it using a machine. And this particular machine is using a technique called capillary electrophoresis. So basically, it's not so much different from the conventional gel electrophoresis that we are using in the lab. Just that instead of using one piece of gel, we are here using capillary gel. So the difference is, as shown in this particular diagram here, the gel is actually one tiny line, okay, that contains the gel inside a tiny line. So the reason why we're using this tiny line is that we can separate the DNA fragment better in a better resolution. So when we're talking about better resolution, we are talking about is able to separate up to single base difference. So if you have 101, the fragment, you can separate it from 102. Okay. So next, we will detect the fragment going through the capillary electrophoresis will detect the signal produced by those fragments. Okay, sorry, a little bit extra on cycle sequencing. Okay, so it's actually very similar to the standard PCR reaction. Okay. However, there are some differences which will be here. Okay. So the first step is still denaturation. We're using high temperature to separate the DNA template. So next, we will have the annealing of a primer. Then we will also have the extension, the elongation of a new strand by DNA polymerase. Okay, so therefore this part here is very similar. The difference is, number one, in addition to the normal use DNTPs, in cycle sequencing, we have additional component called ddNTPs. So we'll look at this in the next slide. Another difference is that when doing PCR, we are using forward and reverse primers. But in cycle sequencing, preparation for Sanger sequencing, we only use one primer in one reaction. Okay, meaning that in one single tube, we only put in one primer, but not two primers. Okay. This also means that because we are using only one single primer, when the new strand synthesis in the reaction, we only get one new strand, but not both new strands. Okay? And the new strand will not serve as a template for the next reaction. Therefore, the whole thing, the new strand increment is in linear multiply, not exponential multiply in terms of the numbers. Okay? So this is showing that after the reaction, we get many short fragments. Excuse me. So this is slide is to show you that the extra component that I was talking about, which is the ddNTP. And as you can see here, the differences is that the ddNTP does not has a oxygen group. This is a normal DNTP. Okay? So ddNTP will remove the O, and ddNTP without the O will interrupt the extension process and generate fragments with different length, so as shown here. So meaning that whenever they acquire a ddNTP, the fragment extension will stop here. So therefore, we have different sizes fragment generated. And then in one single fragment, there will be only one ddNTP. Okay, so and each ddNTP for G, for U, for A, and for T, for C, will be labeling with different color. Okay, so therefore, if you see here, there are different color. Okay, the total difference of color will be just four different color over here. Okay, so after the reaction of cycler sequencing, we will actually move on to dye terminator removal. So the traditional method will be using the ethanol precipitation. Alternatively, we can use purification kit like gel or surfadex or membrane for purification. We bind the DNA. Also, we can opt for magnetic purification. So the DNA will bind to magnetic beads and then elute after washing. After cleanup, now we need to separate the DNA, new chain of the single-stranded DNA based on size. Okay. So single-stranded DNA from the cycle sequencing reaction are separated by the capillary electrophoresis. So as usual, the shorter DNA fragment migrates faster and move past a laser. And the laser will actually detect the fluorescent dye at the end of the ddNTP. And then the fluorescent signal will be detected. As a result, we will produce a electrophoregram. So number 180, we have a T. 181, we have T. 182, we have a C, so on and so forth. Okay, sorry. So next I shall share on the sequencing result, some of the common sequencing results that we've seen, good and bad. Okay, so whenever after the sequencing service has been completed, so we will actually send back 2 files to you, to the user. Okay, so the first file is called a sequence file or txt file, and the second one will actually call .ab1 file. For the txt file, you can just use a normal notepad or Microsoft document or any document program to open it. But once you open it, you will see all those ATCG or N. For the AB1 file, if we would like to open it, we will need an additional software. Okay, so the first one is the ChromaSlide. Okay, showing here, you need to download it and install it. The second one is called Sig Scanner 2. So also shown over here. So the ChromaSlide is the more basic version, and the Sig Scanner 2 is actually a more advanced version of the software to open AB1 file. Okay, so once we open the file, we will see individual peaks like this. Okay, so the individual peaks the important thing that we're looking here is we want to see individual peak, okay, not overlapping of peaks, and very little things below down there. Okay, so when we have something very low over here and we have high individual peaks, we call it high signal to noise ratio, and it's a very good result. Okay, so this is an example of a very good data. Okay, so next, sometimes we will have individual peak. We have some slight overlapping peak. Okay. And we also have some low amount of background noise below. So this depends on your application. If you just want to confirm the sequence accuracy, this data might be still usable. So the reason why we actually observe such events, meaning that there are something below. Okay. One of the possibility is that the signal to noise ratio that I mentioned in the earlier slide, signal to noise ratio. Signal is actually the result of the our new strand of DNA base. Okay, if you go back, so let's say this particular fragment will generate a T signal, a peak of a T here. If there are a lot of this particular fragment, the signal will be high, and then you generate a very high peak. For example, like this G base here has very large number. The number of this particular fragment is a lot. So therefore, we have a very high signal peak. And what is the noise peak? The noise peak is actually not due to our cycle sequencing event. It's something that generated when nothing, even though nothing is there. For example, we have the laser detector. Let's say we did load water, okay, that contains no DNA, no fluorescence into the machine. It will still generate this kind of background noise. Okay. So how we one of the example of background noise is in real life situation, when we go to a very quiet place, and if we cover our ears, you heard some noise. You listen and you heard some noise, but you know that those noise that you listen is actually not a real sound. Okay, it's not a real sound detected by the environment, but it's actually a dead silence noise. So this is the example of noise. So in machine instrument, even though when we're doing detection, they have such a noise. So the important here is the signal generated from the cycle sequencing fragment. It has to be high so that we can actually differentiate away from the noise. So for example, here you can see that these are actually the peaks of the noise. Okay? And then whenever we have the signal from the cycle sequencing events fragment is very high compared to this particular noise, okay, we can actually remove those noise will not be seen in the real data. We can easily remove all this noise. However, if our signal peak is very low, very close to the noise, then you will generate this kind of data, where the real signal is hardly differentiated from the noise, and you will still present. Okay, so one of the reason, this is the even worse data. Okay, so if let's say the real condition of this is not very, we cannot determine the real reason of this by just looking at this particular file. However, if we actually look at the raw data of this file, and if we only detect the signal, this is the signal strength. If the signal strength is actually below 100 units, means that the real signal is actually very low and then overlapping with all the noise. And that's why we see very high overlapping of the signal. Okay. One of the reasons why we have very low signal over here, nearly as high as the noise, is because of low template concentration. Meaning that when we perform cycler sequencing, we actually added some template. If the template concentration is too low, we are unable to generate a lot of fragments because the template is very low. And if the number of these kind of fragments are very low, okay, it will actually translate into a low signal. And when you have low signal, okay, the signal will not be able to be differentiated away from the background. And then we have overlapping. Okay, so of course, in another case, if we actually found that the signal is normal, the signal strength is normal, like 500 and above, nearly 1000, but we still see this kind of overlapping, then the possibility is that it might have a multiple template. It might be caused by some other reason, like specific application or some other reason, but not, let's say, low template concentration. Okay. So another case that we will see here, we have good data. So before 190, we have good signal data, very individually picked, and the signal very high, low background. But the thing is, after this particular point, we observe some overlapping result. It's no longer individual peak. So this one actually can be caused by this particular region here. Okay, we call it the poly region. So this particular poly T region caused something that we believe to happen, which is called slippage. So this one shows that the possibility of how slippage actually happened. Okay, the T here actually caused some loop or hairpin, and then that's why caused an additional T or lacking an additional T. So therefore, you see, instead of a T, we see the next one over here, or we have an additional C over here, and therefore the signal has been like overlapping and overlapping due to the slippage and also the generation of additional and also lacking one base. Another event, okay, we have good signal at the beginning, but the signal drop very fast. This is actually a normal graduation decrease of the signal when we look at the raw signal data. However, in some cases, it's like going downhill. You're like seeing a signal decrease like a slope. So if you see a slope, okay, then it's actually not very normal. One of the possibility of seeing a slope, okay, of this dropping of signal could be explained possibly. The one of the reason is presence of sorts of inhibitors. Okay? the inhibitor might be killing the polymerase, causing the polymerase no longer generating the longer fragment, and therefore the numbers decreasing, and that's why we have very little signal in the end. And also the salt will actually causing some issue and cause the signal to drop earlier. And in this another case, everything of the signal in the beginning, okay. And then up to a point again, we have overlapping sequences. So this is different from the case that we looked at earlier, where we have some either poly T, poly A, poly G, or repeats, AT, AT, a long fragment, long stretch of repeats, AT, 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 or so on and so forth. We didn't detect any of this kind of arrangement over here, so that's why this is a different case. Okay. And the weird thing over here is that we can only see two peaks. Okay. Each peaks over here, we can only see two color. Means that there is only two information available. So this is possibly caused by an allele with deletion or insertion. Means that one allele we have the normal copy, the up there one. Another allele we have some deletion, that's why it cuts short. So the signal actually move forward. So therefore an overlapping. Okay. So after looking at some of the good results and bad results, here I will share on some sample preparation steps. Okay. So at the end of PCR reaction, a lot of time we will send the PCR products for sequencing. So we will need to remove the reagents inside the PCR reaction so that we can continue with Sanger sequencing. So when we perform the PCR cleanup step using the cleanup kit, we are actually removing polymerase, which is actually a protein. There will be remains of the single-stranded 4' primers. Okay, as mentioned earlier, we only have one primer in the cycler sequencing, and therefore we need to remove all this excess primer from the reaction first. Next, we will have excess of the DNTPs as the remains of the PCR reaction. We need to remove that. We need to remove the chemicals, buffer, and then to change the pH and all that. Okay. So when we're using a PCR cleanup kit, so we can actually increase the concentration by eluting with a smaller volume. Okay. So as mentioned earlier, one of the problem of DNA sequencing that we encounter is we have low sample concentration. Okay. When we have low template concentration, it might actually has an issue to generate high enough peak and have a good signal to noise ratio. For that reason, we can actually try to concentrate our sample after cleanup by using a smaller volume. Okay. And then some kit like the Monarch, we are actually able to prevent buffer retention and prevent salt carryover. So this is also help for the second issue that I mentioned earlier, where salt carryover might actually cause the early signal dropping. Okay, so the slope effect of the signal dropping and causing the DNA sequence to end earlier. Okay, so also for Monarch kit, we actually sell the buffer and column available. So if you just finish a buffer, you can just buy the buffer separately. If the column is finished but you have a buffer, you can just buy the column. So another type of cleanup, so this particular one, we are using a column. Okay, so we have a column membrane here and it will bind the DNA, and then we actually wash it with wash buffer, remove all this unwanted thing over here. Okay, and then after that we actually add the elution buffer to actually elute the DNA away from the membrane, and we actually collected the eluted DNA. Okay. Here we have another type of cleanup kit called ExoCIP. Okay. So Exo is actually an exonuclease, an enzyme, and CIP is actually alkaline phosphatase. So the principle of this particular kit is to rapidly degrade the residue PCR primers and then dephosphorylate excess DNTPs. Okay, so meaning that whenever we have the excess primer, exonuclease will digest it, okay, into DNTP. And then for the DNTP, we actually remove the phosphate group so that it cannot become the substrate of the extension event. The good thing about the EXO CIP kit is that the reaction will complete in just 4 minutes, so very fast, and it can be heat inactivated. The only downside of this particular kit is that it can only digest single-stranded DNA, for example, the forward primers and the reverse primer. However, if the PCR reaction actually generate a primer dimer, okay, so whenever we have primer dimer, it will actually form a double-stranded unspecific products. And because it's double-stranded, the exonuclease will not be able to degrade the primer dimers. It will be remains there. So for, let's say if you have a primer dimer issue that causing your problem to your Sanger sequencing, or your PCR product is actually consists of multiple band, okay, then we can opt for using the gel extraction kit. So we will cast a gel as normal, we'll load DNA inside, we separate bands with different size, and then we cut the band contained in the gel. So one of the things over here is that whenever we cut the gel, we try to limit the size of the gel. Okay, because when if you are cutting the gel to become too big, the size of the gel is actually, when we do the cleaning step, we might not be able to remove all the gel components. Okay, so therefore it will be a problem if the gel size that we cut is too big. Okay, so next, some tips and tricks for sample preparation. Okay, so the first important point is that when we elute the DNA from, let's say, the cleanup kit or from the gel extraction kit, elute the purified product or plasmid with nuclease-free water. Okay. Because sometimes when we use elution buffer, the elution buffer might contain EDTA. So EDTA is actually some ions that might cause a problem to the signal detection and also the electrophoresis, the capillary electrophoresis. Okay, so TE or any reagent with EDTA might interfere with the cycle sequencing reaction and generate noise data. Okay. The second thing is we can, before we send the sample for sequencing, we can load it to gel and check for the concentration. Okay, so as mentioned earlier, if the sample concentration is too low, so insufficient products will generate unreadable signal. So the signal is too low and then the noise will be overlapping with them. Okay, so a simple start thing is that we're just comparing the signal with the ladder. So this one, the band intensity is similar, so it's very good. This is even better band because it's higher than the comparison band. Okay. Okay. So if you have the option to measure the concentration, then the recommended concentration for PCR product is actually 30 to 50 nanogram for plasmid, more than 100. Okay. If you do not able to measure them in terms of nanogram per microliter, you can just look at the intensity of the band comparing to the ladder. Okay. So the next thing is we are trying to have a single band. Okay. If we have multiple band, then the signal of both band will be overlapping and shown in the sequencing result. Okay. So for multiple band, we need to do gel extraction. And sometimes if the primer dimer is too much, it might cause problem as well. Okay. And certain cases, a lot of times whenever we have a customer doing 16S primer sequencing, okay, using the 27F and 1492R, okay. So the sequencing result a lot of time we also has what we call early signal loss, okay, meaning that after maybe the full length of the 16S is like 1,500, but the sequencing reaction will show you the result around 5 to 600, and then the signal after that is actually very hard to have good signal strength, and therefore we don't know what's the base be after 500. Okay. In those kind of cases, maybe we can consider using a different pair of sequencing primer, for example 785F and also 907R. Okay, so it's actually used for the sequencing of the same PCR product generated by 27F and 1492. Okay, but the 700, 800, 785 primer, you bind at the center of the PCR product and then move on. And then the 907 will actually bind near to the center, okay, and then move backwards. The result of these two will cover the full length of the 16S. Okay. Their binding site is not on the 27 and 1492. Sometimes the reaction problem of this sequencing might be caused by the binding of 27F to the PCR product. Okay, so that's why the alternative is use a different sequencing primer for the product of 27 and 1492. Okay, so after we get the sequencing result, a lot of time we just need to check the sequence accuracy. Confirm the sequence by using blast tools from the NCBI. Okay, so it just actually binds region of local similarity between sequences, and then we compare to the database. Okay, so either the nucleotide or the protein. Okay, so before I ended, so this will be a little bit about the advancement of the sequencing technology, which is actually the next generation sequencing, NGS. Okay. So compared to Sanger sequencing, a lot of time we use PCR to generate the template for sequencing. In NGS, we do not need to use PCR, because PCR we need to have the primer sequence and all that. In NGS, we just need to break the long DNA fragment into shorter fragment, and then we sequence all these short fragments, and then we join them together into a single unit by using bioinformatic computer. So these are the application of NGS. So compared to the Sanger sequencing, it can have more usage in different fields. Okay? For example, full genome sequencing, RNA sequencing, and metagenomic sequencing, and so on and so forth. Okay, so as a summary of today's sharing, okay, so Sanger sequencing is very good if you want to sequence targeted fragment, just a short fragment and it's targeted. Whereas just now NGS, you can sequence everything. Sanger sequencing is more cost effective if a short region is your interest. If you want to sequence a very long DNA fragment, then go for NGS. And Sanger sequencing provides valuable information for the study of genetic variation. However, bear in mind, the take-home message that I would like you all to have will be the sample quality is the most important thing in Sanger sequencing for it to be success. Okay, so with this, I will wish that everyone will get very good sequencing result for your project. Thank you.
Thank you, Chi-Hong, for the wonderful presentation. Now we will go for the Q&A session. Please feel free to voice out any question by unmuting your mic or type your questions in the chat box. So I see that there is one question in the chat box from Lee Rong Chan. What are the DT-NTP bases and method for dye terminator? Chi-Hong, could you please answer?
Okay, so I will share back my screen so that I can show you the slide. Okay, what are the DDNTP phase and method for dye termination? Okay. 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 So actually, over here, one moment, my pointer. Okay. So you can see that this one is actually a normal DNTP. So this one will be used in the normal PCR. So in a normal PCR reaction, we want to extend the sequence. Okay, we have this normal one, and this normal one also existed. I mean, we also have this in the normal cycle sequencing. Okay, because we want to produce new strand, we need the normal nucleotide. But in cycle sequencing, we have one additional nucleotide that is called DDNTP. Okay, so in cycle sequencing, we have the mixture of these two. Okay, we have a certain ratio. Okay, maybe 70% of this, 30% of this, depending on the length of the template that we would like to sequence. Okay, so the difference between the DNTP and the DNTP is that the chemical group over here. Okay, so you see that if let's say imagine now I have another DNTP. Okay, so I want to join to the next nucleotide. So the reaction will happen down here. The phosphate group of the next DNTP will actually form a reaction with the OH group here. Okay, so if you look at the DNA chain, okay, you see that there is a phosphate group here, and then a phosphate group from the next base is actually joined with a O. Okay, originally here is the OH. Okay, so OH with the phosphate group will form a chain. Okay, then we form the phosphodiester linkage. So that's why the next base, okay, the phosphate group and OH. That's why the OH here, the O is essential for the linkage. If you would like to join a next base to the original chain, you need the OH group to be present in this particular base. Okay? Okay, so we need this for the phosphate group of the next DNTP to form the reaction and form the diester chain. Okay. However, by random, okay, if let's say the chain at DNTP, 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 and then by chance they pick up the DDNTP, so you added the DDNTP, but the DDNTP do not have a OH group here. Therefore, the next base cannot be added below here. And because this event is random, okay, because we have different ratio of the combination of these two, therefore we will generate fragment with different sizes. This particular fragment able to add 1, 2, 3, 4, 5, 6, 7 DNTP before it able to pick up a DDNTP. And because it pick up a DDNTP, therefore the next base cannot form a bond with the H group here. So therefore, it will no longer be extended. It will stop just here. So therefore, for all this, it will just stop just here. And we have different sizes, and that's for that reason. Okay, so therefore, after the cycler sequencing, we actually have fragment with different sizes, and this one, DGTP, we have different color. Okay, so this one is a TTP, this one is a CTP. Okay, so there will become a mixture of all the fragment with different sizes and with different dye labeling. Of course, because it's in a mixture, we are unable to know which one comes first. Okay, so therefore we actually loaded this thing into a capillary electrophoresis system. Okay, and because of the capillary, we are able to move and separate sequence with, let's say, just one single base differences. This one will move first. Okay, we'll pass through the detection of the laser first. So we detected T. Then this one will come in second. Okay, then the next detection of the laser will become a T. So this one come in number 3, the third fastest, and then we detect the laser, we detected a G signal because of the coloring, and so on and so forth, because they are actually movement with different speed based on their size. Okay, each one in turns pass through the laser, and then will be detected the base, and therefore we have this kind of result. So this one you can imagine, this one is 180. So you can imagine there is a fragment with 180 with size ended with a T here. This is the fragment with the size of 181, okay? And the last base is actually a T. And of course, when we're talking about this fragment pass through, it will not be only a single fragment. There are maybe 500, 1000 copy of this particular size fragment with a single T there. Okay? There are many copies of this particular fragment with this size. All of this that ended with a T. Okay? And the number of fragments here is actually important to generate the signal intensity. Okay? So the peak height is actually determined by how many number of this particular fragment ended with T over here. Okay, so I hope that my explanation is clear.
All right. And we have another question from Min Lim. Hi, how to increase the concentration of the gel extraction? What is the suitable concentration of gel extraction to send for sequencing?
Okay, thank you for your question. So unfortunately, to me also, gel extraction actually will more often to cause problem compared to PCR cleanup. Okay, one of the reasons is because the recovery of DNA fragment from gel is actually lower than normal PCR cleanup. Okay? So for normal PCR cleanup, maybe we able to recover 90% or 85% of the bands in the that we actually loaded. Okay, so we can easily recover most of the PCR products that we actually want to clean up. But for gel, okay, sometimes the efficiency on binding and all that is actually lower. We might actually lose more DNA fragment when we perform gel extraction. So therefore, in order to increase the concentration, okay, so one of the suggestion is to actually elute it with less elution buffer. So like for example, certain kit, they will give you a range of how much solution you can actually elute. Okay. If let's say you can only elute it with 50 microliter, okay, compared to another kit, you can elute with let's say 10 microliter. Of course, the concentration of the 10 microliter one will be higher. Okay. When you elute with more elution buffer or water, the concentration of the product will be dropping. So therefore, one of the trick over here is try to see whether the kit can handle it. If they can handle low amount of elution volume, then this is the easy way. Otherwise, then we might need to pull up some to pull up the PCR product. Okay, we try to pull up more reaction of a PCR product, and then hopefully the well is big enough to fit in more than one reaction. Okay, or in certain cases, we might also consider okay, we actually have side by side. Okay, let's say side by side. Okay, let's say this one, sorry, point. Okay. Let's say this is the same reaction as this one. Okay, so we actually excise this particular one. Okay, and then we excise this particular one. Okay, we actually heat it in for melting and all that. But when we load, we actually try to see whether we can actually load both reaction into the same column. Okay, so instead of just using one gel size, okay, so if the well is not big enough, we try to see whether we can load both reaction into the same column. However, there is a risk when we're doing this because we don't know whether the membrane can handle so many gel or not. Okay? Or after we purify using gel, okay, two reaction, okay, then we actually do additional purification using normal PCR, okay, but we combine two tubes of the gel extracted band. So we perform gel extraction first. We got two tubes, and then we combine it using, let's say, a PCR. Okay, so hopefully my comment is useful.
Right, thank you, Chi Hong. We have another question from MY Chin. What is the reason the sequencing failed completely?
Oh, okay. One of the reasons that the sequencing failed completely, okay, meaning that, okay. So when we're talking about the reaction fail, a lot of time we are talking about cycler sequencing reaction fail, okay? Means that we only see some peaks around 20 or less than 20 over here, okay? We see the peak signal, the signal is high, not okay, but then after the around 20, it dropped all the way down, nothing to the background, okay? Drop to here. Okay? You see something like this, okay, high around 20, then suddenly after this one, it's straight away you see the signal become like this. So this is what we call a failed sequencing event because there are nothing there. Okay, and a lot of time our understanding is that the template will be an issue because there are no template, and that's why we are unable to generate any this kind of short fragment at all. And second thing is possibly not common, but when we do sequencing that time, the primer is actually not the same one. Okay. Let's say the user sent us the sample. They told us that we are using the universal primer 16S. So please use your, because we are sometimes for those universal primer, so sequencing house will actually use their in-house primers. Okay. So when they use the in-house primer means that the user do not need to send their own primers. Okay. So that's why in one cases, the customer say, oh, I'm using this particular primer. So please use this for sequencing. However, the problem is we didn't check the sequence similarity between the one that he used for PCR and the one that we in-house having. Okay? Although the name is the same, but unfortunately there are differences in terms of their bases. And therefore, the primer binding is no good. Therefore, the cycler sequencing event failed. Okay? So, however, this one normally is considered a rare event because we can easily check the gel to see whether the template got template or not. Okay, because when we load the DNA here, if we hardly see any band, okay, it could be we will form very big problem because if the concentration is so low until undetected on gel, very likely that the cycler sequencing event will fail. Okay. Another thing is we understand that customer will actually check their PCR product in their lab and all that. Okay, but the problem is for some reason in certain cases, although the band looks okay in the user lab, maybe during transport or something happened, maybe degradation or whatever happened. So when it reached the sequencing house, the DNA may be degraded, and that's why when we load it on the gel, we do not observe anything. Okay, so that's why although we are quite convinced that they are sampled, but unfortunately when reached the sequencing house, something happened, the band no longer there. Okay, of course when we load this like very low concentration template there, the cycler sequencing event will fail.
Thank you, Chi Hong, for the answer. So, is there anyone have any question for our speaker today? You may unmute your mic to voice out your question.
Okay, actually I do have. Hi, Chi Hong.
Hi.
Okay. Our sample, right, is actually single-stranded DNA.
Okay.
Okay, I just want to confirm with you lah. Okay, ours is single-stranded DNA, and then we send 2 primers over.
Okay.
All right. So the result that we got, right, we got back is actually one of the primer gave us very good sequencing. The
Yeah.
other one is actually a lot of noise.
Mm.
So
This
that
is
is
normal.
okay. So that means our sample is not enough, right?
No.
No.
My understanding is because you are sending single-stranded template. Am I
Uh-huh.
correct? Yeah.
Yes.
Yeah. That's the reason why you can only have one sequencing event when you have a single-stranded template.
Oh, but we gave 2 types of different primers.
Yeah. And if I understand you correctly, okay.
Ah.
Okay. So I try to show you. Okay. Okay. Let me go. Okay. Let me go to this one. Okay. So if I understand you correctly, you only have one type of sample. That one type of sample is actually single-stranded. Am I correct?
Yes.
Okay. Let's say this is please ignore everything on top. Okay. Let's say this is the single-stranded template. Okay.
Okay.
Okay. So you can see that it is 5 to 3.
Hmm.
Okay. So therefore, sequencing event can only happen for this particular primers that actually moving from 5 to 3 sequencing to happen here.
Mm-hmm.
The other primer that actually starting from, let's say, this direction, okay, it has nothing to bind.
Oh, but we are giving 2 different reverse primer.
Oh, okay. So means that one over here and the next one over here.
Ah.
In this case, that because one sequencing event actually success, I think that the next one should be success as well.
But
So
yeah, this is what we get. One of the reverse primer is actually good, but the other one has a lot of noise. So that's why I just want to confirm whether is it our sample not enough.
because one of the sequencing event show that the template is okay. So
Mm.
if we want to double confirm, then you can use the what we call the six scanner software. Okay. Then after that, you open the raw data signal.
Mm.
So from there, you will actually see the raw signal strength. So raw signal strength, or if you have low sample, the raw signal strength normally what I see is actually lower than 100.
Yeah. So it's
So
so
if
that means
let's say your first sequencing, the one that success, it has high, signal strength more than
nice.
100,
Yeah.
means that the template is okay. The
Okay.
concentration is okay. So the problem should not be the template problem.
Oh, so
Maybe is the second primer binding problem. I'm not sure
okay.
whether.
If like this, right? Because we are giving like what you said just now, right? Your
Mm,
technology is actually using 1 primer at one time, right? Not
yeah.
so, which means that when we give you 2 reverse primers, means you will do
Mm,
it the same DNA template, but
yeah.
2 times,
Yep. That's why if the
2
first one
separate times,
success and if the first one the signal is normal more than the 500, the
ah.
second one should work because the template is enough.
Oh, so
Yeah.
what do you think is the problem? If one of it worked and the other one didn't work?
I can only guess.
ah.
So because I don't know, are you confirm that the second primer sequence will confirm will bind to the region?
Oh,
Is
yes,
it?
yes, we have sent it before. Yeah. The
Oh,
previous
you sent it
rounds were
before.
okay. Just this round
If
only.
you send it before is confirmed, and then you can request for. I'm not sure who you send it to, but if let's say
Okay.
you send it to us, you can request for resequencing and see whether the same result you're getting
Oh,
the same
really?
result. Ah, ah.
Oh, then luckily I asked.
Uh,
Okay. So, okay.
if
So
there
the
are still if there are still template there, they will actually help you to perform a repeat. Okay, so if the repeat also show that it's not successful, means that it's not a technical problem.
oh,
Then
okay.
maybe some mutation or need to think what really happened.
Oh, really? Yeah, because
Mm.
only this round, because the previous round that we sent were okay.
In that case, you can always contact us and see whether it's possible to do a repeat lah.
Oh, okay. So which means, right, if the one of the primer work, the other one should work as well, lah.
Yeah, because when we're talking about the concentration thingy, since in this case we are using the same template, so the concentration should be the same. So
Oh,
if the first one show that the signal strength is
good.
good, means that we have enough template. So the
Oh,
second one should work.
okay. All right. Okay. One more thing. Because in your method, right, you also mentioned that the first step that you do is actually denature the double-stranded, correct or not?
Yeah.
So in this case, if we send single-stranded, will you also denature?
That I'm not sure, but I think so.
Okay.
Yeah, but it wouldn't affect anything.
Yeah. Because we didn't do the cleanup, actually. We actually, in our electrophoresis, right, we actually see a few bands. I mean, two or three bands. But there was also what happened previously, lah. But because it's
in that case, then maybe you can contact me, or you can let your salesperson know. Then they will send your sequencing result for me to see, because I want to know whether the second primer failure is because of really multiple template, or because it's like I want to see the signal strength and all that.
okay.
Yes,
All
I
right.
will look into it. Yeah.
Okay, thank you very much. Thank you.
Sure, no problem.
Right, thank you. Is there any more question from the floors? Well, it seems like no more question. So, Chi Hong, is there anything else that you wanted to cover before we end this webinar today?
Okay, so I will share my email here. So if you have any problem, you can reach out to me. I will see what I can do. Okay.
Okay. Yeah, also,
Yes, I see somebody raise hand.
hello, is there any more question? No, right? So, yeah, we are going to end our webinar for today. And thank you everyone for joining this webinar. Before you leave, please fill out the survey form as attached in the chat box. Also, we will send the recording of this webinar and the slides to all the participants after the webinar session. Lastly, thank you again for joining the webinar, and see you again next time. Thank
You
you.
everyone for your joining. Thank you everyone for your attention. Have a great day. Bye.